Wonderful. Okay. I regret I can't be there in person today. Having attended probably half a dozen ISMRD meetings in the past and organized the last one in person in Atlanta in 2019, I can certainly attest to the power of all these voices coming together. And I think it's been a wonderful experience. I'm going to start with just giving you an overview of mucolipidosis II or eye cell disease. I'm really going to refer to these as the GNPTAB related disorders. And I want to start with the cell biology that sort of underlies this because it's not only fascinating, but it's integral to understanding these conditions and why they're so unique. And it really start by answering the question, how do lysosomal enzymes or acid hydrolases reach their target compartment, the lysosome? And this is done through a very elegant carbohydrate-mediated pathway where the enzymes are tagged with a sugar moiety called mannose-6-phosphate on their own end-linked sugar chains by an enzyme called glucnac-1 phosphotransferase. And by tagging these enzymes with this mannose-6-phosphate, it allows them to recognize or be recognized by receptors in the trans-Golgi network and be diverted away from the secretory pathway and targeted to the lysosomal compartment. The receptor then will drop those enzymes off, go back, pick up more enzymes. And in this way, the lysosome can be furnished continually with one of 60 soluble lysosomal hydrolases that are needed to degrade all manner of material that ends up there. The enzyme itself is actually quite interesting. It's encoded by two genes, GNPTAB and GNPTG. And the GNPTAB gene product is cleaved into two subunits, alpha and beta. And so you end up with alpha, beta, and gamma subunits, and these come together in twos to form a heterohexameric enzyme that resides in the Golgi and does this job. Now, we thought we understood all of the aspects of this pathway. And before I go further, I just want to mention this was really all worked out by seminal findings from Liz Neufeld, Bill Sly, Stuart Kornfeld, and then after that initial discovery, continued work from Stuart's lab as well as Thomas Brock and many others that I can't name. But we thought we understood the full pathway, and then recently there was a protein TMEM 251 or lyset that was identified by Stuart Thomas and Ming Li's group that is required for stability of the gene products, cleavage by the enzyme that cleaves alpha and beta, and also enzymatic activity. So even with a story that's been so well documented, there were still a few surprises. But what happens, and there's the OMIM entries for those that want to record that. What happens in mucolipidosis II is that glucnac-1 phosphotransferase enzyme is missing. And because it's missing, or if it has defective function in some of the more attenuated forms, there is either no mannose phosphorylation on the lysosomal enzymes or reduced. And now they don't have that necessary tag to be able to be transported to the lysosome. And what this sets up is a number of different consequences. The first being that, yes, you have lysosomal storage because you're missing a whole variety of different lysosomal hydrolases. But unlike other conditions, these enzymes are all still made. They're just mistargeted. And where they end up is actually outside the cell. So they're still made and folded and potentially still active. They're just not reaching their compartment, and instead they're ending up outside the cell. And myself and my wife and colleague, Heather Flanagan-Steet, did a lot of work over the past 15 years on really trying to address the impact of what happens when these enzymes are secreted outside the cell. And our work focused mainly on these proteases, one particular protease, cathepsin K, that was active outside the cell and was capable of disrupting a lot of signaling pathways that are important for bone and cartilage development. But this, I think, highlights what makes this so complex. It isn't just loss of one single enzyme. It's trying to understand the impact of mistargeting or loss of lysosomal content of a whole range. And I think that's an important point, especially when we think about treatments. This one has complexities that make it particularly challenging. So these are the faces of mucolipidosis. You could see this picture of a girl, affected female over here on the left. Obviously, profound skeletal involvement, very coarse facial features. Some of the features, especially what's happening in the face, do resemble other lysosomal conditions like MPS I or Morquio syndrome. The more attenuated form is over here with this affected male child. And you can see maybe the skeleton isn't as affected, but still has the contractures of the hands, some coarse features, and has difficulty raising his arms above his head. So we view these diseases really as connective tissue or skeletal conditions. So what I'm going to move into is a bit of a departure from thinking about therapy and talk about potential mechanisms of drug resistance in this disease. And you might be asking, why study drug resistance in the context of mucolipidosis II or other LSDs? This really came from discussions with the families and what they were seeing with their children, and could we address something? So I think that reinforces the importance of having that communication and hearing what the families and the patients are dealing with. But one of the concerns was families were noting, and again, this is anecdotal, but they were noticing that more and more pain medication was needed for these patients in order to achieve the desired outcome of relieving pain. And this was particularly important or it increased in prevalence as at end stages of their lives. So we set out and said, could we do something to just explore why patients might have resistance to pain medications? I don't know if we've answered that question, but I think through this study that I'm going to share with you, we did highlight some new and interesting concepts that can be pursued in future work. So I'm going to take you through this paper. This was a study that was supported by Cure Mucolipidosis and the Spanish MPS Society and published last year. And this is an open access article, so it should be able to be viewed by everybody. But the way we went about this is we wanted to generate a new ML2 cell line. And so we knocked out, using CRISPR-Cas engineering, knocked out GNPT-AB and a cell line called DAOY. And this is a medulloblastoma line. It has neuronal characteristics, and there hadn't been a neuronal-like cell line knocked out of this gene generated. And so we thought this could be a useful tool. So we did the CRISPR engineering, isolated 2 clones, D3 and E4, and then began to characterize those clones. And down here, you can see the content of intracellular enzyme activity, wild type, and then the 2 clones. And as we would expect for mucolipidosis II, we have really profound deficiency of several lysosomal enzymes, including beta-galactosidase, beta-hexosaminidase, and beta-glucuronidase. But you might notice over here, this curious finding, beta-glucosidase, which is the enzyme involved in Gaucher disease, actually has elevated activity. And that's because this is one of the few enzymes that actually doesn't utilize the mannose-6-phosphate pathway, but instead traffics to the lysosome by other mechanisms. So we went on to do additional characterization of these cells. We showed that they have lysosomal proliferation and storage. They have accumulation of cholesterol, which we assessed by Philippin stain, and they have all the signs of impaired autophagy. So down here in this Western blot, you can see a significant increase in LAMP2, reflecting the fact they just have more lysosomes, an increase in the autophagy regulator P62, which I'll come back to at the end, as well as altered levels, ratio of LC3 to 2, which is indicative of impaired autophagy. So we went on to do some omics level studies on these cells, and one of those was to do RNA sequencing. We looked at the RNA sequencing results. Not surprising, there were several upregulated and downregulated genes. Looking at the gene ontologies to see where these up and downregulated genes were enriched, highlighted some interesting pathways that I don't think we often think about in the context of ML2, like downregulation of arginine biosynthesis, striking upregulation of enzymes involved in cholesterol biosynthesis, which again might be an outcome of the cholesterol storage that is very clear in these cells. And then if you look further down, a number of other interesting pathways that are affected. I'll highlight here Alzheimer's presenilin, further down Alzheimer's amyloid secretase pathway, Huntington's disease. We were seeing a lot of pathways that are relevant to neurodegeneration. And I'm going to build on that a little more in the subsequent slides. So that was all RNA sequencing. That's looking for changes in gene expression. But then we wanted to ask, what is happening actually at the cell surface of these cells? So we did cell surface proteomics using a method that I worked on when I was at the University of Georgia, where you can tag cell surface proteins with a sialic acid, which was discussed in the last session, that has this biotin tag, and it lets you pull down all of the cells that are only at the, or all of the glycoproteins that are only at the cell surface, and then do proteomics and quantify which proteins are increased in abundance at the plasma membrane and which ones are decreased. And I'll highlight here again is this APP. This is the amyloid precursor protein. We also saw an increase of cell surface resonance of the LDL receptor, as well as IGF-2R, which is the cation independent mannose-6-phosphate receptor. These made sense because they're either directly or thought to be indirectly involved with bringing enzymes back into the cell. So it made sense that those were up. But I thought this one was interesting to see an increase in the amount of APP at the surface. We went on to show that there is an increase in the abundance of this protein in the clones. And I think this highlights, again, we don't know the exact role of having an elevated level of this APP protein in this condition. But I think what it reinforces is the growing evidence that altered lysosomal function is relevant to a lot of neurodegenerative conditions. Of course, we have a very good example with acid beta glucosidase and Parkinson's disease, but with more recent work from Mark Sand's lab, I think we need to pay attention to what loss of function in lysosomal enzymes might be doing more broadly to contribute to neurodegenerative disease. So this chart just shows increased and decreased abundance of membrane proteins in these cells. And you can see this one that is highly increased in the knockout cells compared to the wild type is our protein P62. So we had already shown that that was elevated, but and there were a few other things that we thought were interesting that could be related to drug resistance. But really this looking more deeply in the literature at what was going on with P62 highlighted that there is some papers out there that have looked at the impact of autophagy on morphine tolerance. And that's basically, the example that we were trying to understand better. Tolerance to pain medications like morphine. There were a number of papers that really talked about autophagy and altered levels of P62 to morphine tolerance. So these, I think, collectively indicate that modulating autophagy, it may be a reasonable target to prevent morphine tolerance. But I think this at least sets the stage for trying to understand this further. And I think through our study, even though we couldn't find the smoking gun, if you will, for why there might be increased pain tolerance, at least it does highlight and generate a set of tools that can be used for future studies. So that's all I've got. Be happy to take any questions. And I'll be here to weigh in on some of the other discussions.
Rich, thank you for that elegant presentation and really fascinating work. I just had some questions. You mentioned the number of downstream pathways affected. A lot of lysosomal disorders have secondary mitochondrial dysfunction. Did you see other evidence of that in the studies that you did?
Yes, I think we did not look directly at mitochondrial function, but that would be one interesting aspect to explore. I do think some of the gene expression changes are highlighting some alteration in mitochondrial function. I don't know if you noticed, but one of the pathways that was upregulated was glycolysis. That is typical if you have cells that have compromised oxidative phosphorylation and energy production due to potentially defective mitochondria or defective clearance of damaged mitochondria. So I think these are the types of things, resources that I hope if others are aware of them and want to follow up, I think it would be great to see what's happening at a deeper level.
Thank you. Any questions from the floor or from our online audience? Again, very, very elegant work and exciting, particularly with the relevance, I think, to pain tolerance and effects on morphine. I think this is really fascinating. If I can just ask, was this a specific target you were looking for, or was this a case of a prepared mind recognizing a potential link?
Yeah, I mean, it was not. It was going into it, it was not immediately clear why you would be experiencing this, but I actually think the autophagy argument makes a lot of sense that you have some impaired autophagy. We know that certain medications, all drugs, there's a number of drugs that will come into the lysosome and become protonated and then not be able to exit out. And so there's a lot, related to multi-drug resistance and lysosomal function. But what struck me, and I don't know if this is noted in other lysosomal conditions, was just that there is an issue with pain tolerance in some of these patients, particularly towards end of life.
Thank you. I think that's really a great insight. We have a question from the floor. If you'd like to come up to the microphone, ma'am, it's right there.
Hi, Richard. It's Susan Kester, Lonnie's mom. How you doing?
I'm well. How are you?
Good. I'm good. I just wanted to mention that Lonnie has always had a very high tolerance to pain. We've never really even used any pain medication with him other than over the counter, except during his heart surgery, of course. And secondly, he has experienced different side effects from different drugs, especially anti-anxiety and depression drugs that have gone like straight to his heart. Side effects that the doctors that treat him on a day-to-day never thought he would exhibit. So again, we want to thank you for doing this study. And if we do need to go further, we certainly know who to call, right?
Sure. Yes. No, thanks for that. I mean, again, I think that in this field broadly, not just in the glycoproteinosis, but in lysosomal storage diseases in general, we're just scratching the surface on drug metabolism. And obviously, there are more pertinent things to be thinking about for a lot of these patients and families. But I think understanding, how impaired lysosomal function is impacting drug metabolism is an important question to be considered going forward.
Well, thanks again, Rich. That's terrific work and elegantly presented as always. I think now at this point we're ready to move to our next speaker, who's Jackie James, who's president of Cure ML. Welcome to the podium, Jackie.
All right. Well, hi, everyone. My name is Jackie James, and I actually live not too far from here in Illinois. And my daughter, Anna, I guess, let's see, we just hit this here. There she is with me. I'm mother to Anna, who just had her 30th birthday. She has ML3, and she was diagnosed in 1999 when she was age 4. So again, long story with how she was diagnosed. Again, one of these things that went on for a while before we actually knew what she had. At that point, we were told there was no cure. Basically make her comfortable, do what you can. Very frustrating. did a lot of research myself, came across the MPS Society, followed by ISMRD. In 2012, I joined the board of ISMRD, and 2015 was board president for 5 years. In 2021, some of you may know Jenny Noble. She has been a tireless advocate. She has 2 children. One has since passed with ML3 as well. And we really wanted to focus and work along the alongside the MCRN, which is a mucolipidosis, I've always thought it was research network that we were working with, to find a cure, find something for mucolipidosis, because it was constantly falling through the cracks. There is work being done. I know Patty's going to talk about that in a little bit too. So that's exciting. We wanted to do what we could. I mean, one of the things that we did was fund what Richard was just talking about with the drug uptake, to see what was going on with our children. My daughter, for example, if I were taking the drug she was taking, I would be on the floor. I couldn't take the medicine, and she's only 100 pounds. She's tiny. So now I am the president of Cure Mucolipidosis. We have some fantastic board members. You met Dan as one of them. And yeah, living with mucolipidosis, as you all know, it doesn't matter which glycoprotein disorder you have, it kind of sucks. There's a lot of stuff that goes on, a lot of things, hardships that you see your children go through. It's one of the worst things that you can actually experience when you see your child in pain, or you see your child not being able to do something that they want to do. In my case, having to deal with almost like a grief in a way that she wasn't going to grow up and get married and have children. You lose all of that. So it's extremely hard as a parent to be able to see your child deal with this. So we know that currently there are no current treatments for mucolipidosis. We formed Mucolipidosis in 2021. The purpose of Cure Mucolipidosis is to find a cure, basically, whatever we can do to help our children. We have, like I say before, developed certain projects. We partnered with Sanford Cords and developed a registry for mucolipidosis, because up until then there wasn't one. We funded $100,000 to the natural history of the cat colony. We funded $40,000 and partnered with the Spanish MPS Society to support Dr. Richard Steet in research project looking at how ML children use drugs. $209,000 was raised in this last year to go ahead and fund year 1 of gene therapy. And I'm going to touch on that in just a minute, what we're doing. And now we're working on funding year 2 of the gene therapy and raising $192,000 to get through that year. So we basically followed sialidosis. We approached the University of Massachusetts, the Heather Gray Edwards. We had, I think, Matthias, and then there was, oh gosh, I'm trying to think. Hector is actually the person that we're dealing with the most. I'm going to read an email from him with an update on the therapy in just a minute. And it's progressing really, really well. I think one of the things that I've learned since I've been on the board, there was always confusion within the mucolipidosis world, and I've heard this being said this morning over and over again with these diseases, is that mucolipidosis is mucolipidosis alpha beta, and that incorporates ML2/3, which is intermediate, and ML3 alpha beta. It's basically the severity. More severe, less severe. And then there is mucolipidosis 3 gamma, which is a separate subvariant. So the research that we're doing for gene therapy is for mucolipidosis alpha beta, and it should benefit ML2, ML2/3, and ML3. So I just wanted to try and make that clear, because we are having confusion with people asking us, like, is this going to benefit ML2? Or is this just for ML3? It should be for the whole thing. So we now have a hope of real therapy. It's expensive, and it does take a community to make it happen. And we have the most amazing community of patients and parents that are coming together to raise this money. So we've got families out there raising money right now. So we are seeing it come in. Like I said, year two is we're on our way. We're doing pretty well for year two. Cure Mucolipidosis Australia Limited has been formed as an arm of Cure ML, kind of similar to Cure Sialidosis. We're all kind of in the same, that all of the people are on the board, but we have kind of more focused groups. They have just raised $64,000 in 3 months, which has been incredible. So a thank you to Juanita Van Dam and to John Delaney for spearheading that. And then we have families now doing GoFundMe crowdfunding. So everything's looking really good on that. I know I need to be brief. So I just wanted to read, I got an email through updating us on the happenings of our gene therapy. So this is from Hector, who's our person at UMass that's doing all this for us. He's like, thanks for checking in. Things have been moving along steadily on our end. Here's a quick update. We successfully cloned the ML2 native promoter together with the GNTB gene, which was a key milestone for us. The mouse tissue samples treated with the capsids of interest have been shipped out for histological sectioning. And I should say we've been able to get mice from the UK, and then Dr. Thomas Brolke in Germany very kindly has given us his ML3 mice. So that's a really big deal. And I believe that just arrived at UMass. Bone samples are currently undergoing decalcification with EDTA, which, as you know, is a slow process. We expect them to be ready for sectioning in the second half of August, so coming up soon. The UK mouse colony has had its first litter, and we anticipate that some pups will be carriers. We're currently genotyping around 100 pups to identify candidates for the next breeding round. Over the next few weeks, our plan is to finish genotyping, amplify the new promoter of interest. And again, this in parentheses, EF1A, scientists probably know this, via PCR for the next cloning steps, and then produce ML2 plus GNP TAB plasmids at sufficient scale for packaging. Once all histological sections are ready, we'll begin optimizing our, and again, this is another scientific word I'm not very good at, immunohistochemistry protocol to evaluate the capsid's ability to transduce both bone and CNS tissues. Finally, we're still actively screening alternative capsids with higher potential to simultaneously target bone and CNS. And that's one of the other things that I really love about this potential therapy is that they are working to include bone in the therapy itself. And as we know, we'll probably go over the outcomes a little bit later. Bone is a really big deal. My daughter has suffered so much from bone pain, abnormalities. So to be able to have this encompassed in the gene therapy is going to be huge. But we all know that ML, mucolipidosis, is a very tricky disorder. It's not quite as simple as the others. And we firmly believe that, you know, working together in community, we may see it may end up being like a cocktail of different therapies that will treat, you know, specific things, or one might be better than another for a certain person. So we're looking forward to walking ahead, and you know, we're interested. We're going to be posting regularly on the updates from the University of Massachusetts. We're so grateful that we're now in this position, because if you'd have told me this 5 years ago, I would never have believed it. So it's been amazing.